mode.
So good morning or good afternoon, everybody. Welcome to this webinar session about quantitative imaging of living biological samples by PeakForce QNM atomic force microscopy. Thank you for joining. We have a lot of attendees today. This webinar is hosted from Europe, so there will be only one single session, but this session is being recorded and should be available for download on the Bruker website in most likely a few weeks. So usually we don't answer questions during the presentation, but you can feel free to send me questions at any time. I have a kind of popping up window where all questions are being collected, and I will try to answer most of them at the end of the presentation. So if we're running out of time, I will anyway answer them offline and can possibly contact you individually by email if needed. But typically we gather all the questions together, we make a Word document, and we send it to all the attendees. I also would like to mention that we have online John Zaraticus, who is our web marketing specialist. There might be a slight delay with the internet connection, so I will try to speak slow, and I will make a short pause between the slides. I also would like to say that we have what we call an exit survey. I will remind you this at the end of the presentation, but please take just 30 seconds of your time to answer the questions. This will help us a lot improve the quality of our webinar presentations. So I would like to go to the next slide, but for any reason it doesn't seem to work. Oh, that's okay. It's a bit slow. So I will start with the key question, which is why force measurements are so essential, so important in biology. Because not always, but sometimes the mechanical properties of cells can be a good indicator of the health of the organism from which they were extracted. The actin cytoskeleton is especially important because it forms a physical connection, a kind of barrier between extracellular matrix, the adhesion complexes, and the nuclear structure. Not only actin, but actually a lot of molecules like cellular adhesion molecules, caderins, tropomyosin, and many proteins involved in the phosphorylation cascade in this pathway play directly or not a role in the cell architecture. So any change or impairment in that organization can lead to severe troubles like development of cancer or possibly neurodegeneration. Generally speaking, a change in local stiffness can be the indicator of a problem. For instance, it has been shown that some cancer cells are usually softer than the normal homologs. Not always, but this has been clearly proved for different types of cancers like breast, colon, prostate cancer, or certain forms of brain cancers. So now you understand why AFM can be so interesting. AFM is a non-invasive technique, depending on what we call non-invasive, of course, but compared to most of classical approaches like surgery in the case of cancer, it's really a technique of choice to investigate ex vivo cells' mechanical properties, for instance, in response to drug treatment, or correlate them with aging or pathologies. So the next slide summarizes how AFM can be operated in physiological conditions, or I should say near physiological conditions, because the fluid in the human bodies are very complex and very hard to reproduce. You have different types of fluid cell, allowing working under fluid environment without any compromise on the FM performance. So the one shown on the left part of the slide is the multimode fluid cell, which was developed about 10 years ago. This was, I think, the first type of perfusing system, and I think it's really good to image DNA of proteins, for instance. And the one on the right side of the slide shows the latest model, which is called the PSI, perfusing stage incubator. And this one is 100% compatible with glass bottom Petri dishes, especially 50mm dishes. So this is how it works. Basically, you have on the top of the dish a first plastic holder, this black stuff, that connects to the second holder, which is magnetic. So this one is attaching the system to the XY stage, the FM stage. And this one is equipped with inlets and outlets for gas and liquid. So normally when you work on living cells, gas will be a blend of oxygen and carbon dioxide, and liquid can be culture medium or any mixture you can use to work on the cells. On the top of this you have a silicon seal to work in a fully closed environment and of course limit the evaporation. So the goal of that, the purpose of that design is that you can keep cells alive for an unlimited period of time. And actually we carried out some tests and we could keep cells alive for 48 hours. So before we jump to the main topic of the talk, which is PeakForce Tapping and PeakForce QNM, I would like to remind you the main differences between the existing FM techniques to measure mechanical properties of the samples. So the first one is tapping mode. This was developed in 1993, and on this slide is shown the principle and also the general description of AFM operations. So some of you might not be familiar with what AFM is. So to make it easy, you have a sharp tip, which is about 1 or 10 micrometers in height, a tip radius of 20 nanometers, which is located at the end of a mounted cantilever on a chip. So this cantilever is about 100 or 200 micrometers in length. And the tip is scanned over the sample surface, which is never perfectly flat. So a laser beam is sent to the back of the cantilever, which is normally reflective. It can be coated with aluminium or gold. And as soon as the cantilever gets bended by the change in topography, the laser spot will also be deflected, and its position on the photodetector will be changed. So this is how you can generate a 3D profile of the surface. So the primary goal of an FM is to generate topographical data, but not only, as we will see in a couple of minutes. So in the meantime, you have a feedback loop to maintain constant position of the force between the tip and the sample. And in tapping mode, the tip is oscillated close to its resonance frequency, so typically 300 kilohertz in air and about 10 kilohertz in liquid, and thus only intermittently contact with the surface. So in that case, the feedback is based on the amplitude of oscillation, which is high when you don't touch the sample and which is reduced when you reach the surface. So the main benefit of tapping mode is that you have negligible friction or shear forces, and among the signals that can be collected other than topography, you have what we call phase signal. So as you can see on the right part of the slide, the phase is the lag between the electrical signal sent to the cantilever and its response in terms of frequency. So depending on the material's properties and the FM parameters that are defined by the user, this phase can be more or less important and will reflect the energy dissipated between the tip and the sample during each tap on the surface. So depending on a parameter that we call ratio set point, which is actually the amplitude set point out of the free amplitude of oscillation, depending on this parameter, the phase signal will be more or less sensitive to surface or volume properties. So for instance, for high ratio set points, this is what we call light tapping conditions, it will be more sensitive to surface forces, mainly adhesion forces. For low ratio set points, this is hard tapping conditions, it will be more sensitive to topography. So if you operate at low ratio set points, you have to be very careful because you're likely to indent into your sample, damage the sample, or contaminate the tip. And for moderate ratio set point, will be more sensitive to volume properties, which are mainly elasticity or viscosity. But most of the time it's not that easy. It's a contribution of several factors, and in any case they cannot be quantified. So this technique cannot really be used to probe cells' mechanical properties. So the second technique is called force spectroscopy. So to understand what this is, you need to have a look at this force distance curve. In that case, it's not really an imaging mode because the tip is brought in contact with the surface. So to make it clear, on the y-axis you have the force in nanonewtons, force between the tip and the sample, and on the x-axis you have the tip sample distance in nanometers. So when the tip is brought in contact with the surface, you have first the contact points, and then it will start indenting into the sample, causing the cantilever to be deflected, until the turn-away point, where the tip pulls off and reaches back to its original position. And from the retraction curve can be detected the non-specific adhesion between the tip and the sample. So this is actually the step height you can see on the red trace, and possibly the specific and binding events if the tip is functionalized with a molecule of interest. So this is about the retraction curve. From the extension curve can be estimated the stiffness by knowing the deflection sensitivity in nanometers per volt and the spring constant in Newton per meter. So this can be applied to single force measurements, which means that you will do this on a single point on the surface, or you can do what we call force volume, which means that you will repeat this on a matrix of points at different resolutions defined by the user. So this technique can be considered as quantitative, but not directly quantitative, because the Young's modulus cannot be directly extracted. The force curves have to be exported, and the Young's modulus can be calculated from the slope of the extension curves by using different types of softwares. Moreover, this technique is very slow compared to FM imaging modes like contact mode or tapping mode, which can be a problem when you work on living cells. And the consequence is that it suffers from a lack of resolution. And as I said, it's not very informative. You can only extract adhesion and stiffness. So now I would like to acknowledge you with a technique, a new technique some of you might be familiar with, which is called PeakForce Tapping, which has little to do with tapping mode and can be understood as a kind of combination of tapping mode and force spectroscopy, in the sense that the probe is oscillated but at far below its resonance frequency. So it's no more 100 kilohertz or even 10 kilohertz, but 1 or 2 kilohertz depending on the FM tool. And the force curve is generated and captured each time the tip taps on the surface. So it works with most of standard cantilevers, not like in harmonics, which is a kind of ancestor technique where you had to use specific cantilevers having a hammerhead shape and a tip offset, because in that case you had to correlate the torsional motion with the vertical motion of the cantilever. This is no more required. In theory, you should be able to use any type of cantilever, of tip. So by standard, the drive amplitude is about 300 nanometers, peak to peak. The imaging feedback is based on the maximum peak force of the force distance curves. And to illustrate what peak force is, maybe you can have a look at the schematic showing the different force fields experienced by the tip during an approach retract cycle. The Z position being indicated by the dashed line, the black line on the top. So in A, the tip is far off the surface and goes through an attractive field first until it reaches the contact point in B. Then it will start indenting into the sample, so this is a repulsive field, a positive force, until the maximum peak force, which is defined in C, and this is actually this value which is used for the feedback calculation. This is what we call peak force. Afterwards, the tip starts pulling off, so you have a maximum adhesion in D, and reaches back to its original position in E or A, which is the same position actually. So the bottom cartoon shows the different states of the cantilever, different bendings from positions A to E. So before I go further with the presentation, I need to define a bit and clarify a few terms. PeakForce Tapping is actually a combination of two features. One of them is called Scan Assist, which is fully automated AFM. So with Scan Assist, which is based on the PeakForce Tapping technology, all parameters like the scan rate, the gain, or the PeakForce set point can be automatically adjusted by the software or individually turned on or off. So I would say that this mode is of particular interest for FM beginners who don't want to waste too much time with parameter optimization. And the second feature is called PeakForce QNM, so QNM standing for Quantitative Nanomechanical Characterization. So the bottom cartoon shows a typical force curve, well, not a real one, but a simulation, that is generated during each tap on the surface, and the information, the signals, that can be extracted. So as in regular force mode, the adhesion is detected as the maximum step height between the baseline and the maximum adhesion, which is the minimum force. The deformation is obtained by the distance on the x-axis between the contact point and the turnaway point. The Young's modulus can be directly obtained by extrapolating the linear part of the curve. So I have to precise that this is a DMT fit, a DMT calculation. I don't have time right now to detail what this is, but I can answer any question related to this at the end of the presentation. The dissipation is obtained by integrating the area between the two curves, and which is missing actually on that slide. The peak force is the maximum distance between, on the y-axis, between the turnaway point and the baseline. And if the probe is calibrated prior to the experiment, I will explain you in a few minutes how this works, all those signals will be directly quantitative, which means that the adhesion and the peak force will be in newtons or nanonewtons, the deformation in nanometers. The Young's modulus will be in Pascal or kilopascal if you work on soft samples, and the dissipation will be in electron volts. So there are two main ways to calibrate the probe in PeakForce QNM, a relative method and an absolute method. So first, for the relative method, you have to engage on a very stiff sample, like a piece of mica, silicone or quartz or glass, which has to be very clean, and you have to update the deflection sensitivity. So this can be done directly from the PeakForce window. You have a clickable button, or even better, if you want to be very accurate, by ramping, capturing a force curve, select the right segment, linear segment on the image, and then click Update Deflection Sensitivity, which will be nanometers per volt. Then you have to withdraw and calculate the spring constant in Newton per meter. So this can be done automatically by using thermotune, which is a kind of sweep on the range of frequencies. So you will find the main peak, you will feed the data, and the software will automatically calculate the spring constant. So next step, you have to image a reference sample with a known Young's modulus and adjust the tip radius to make the measured Young's modulus equal to the known one. And the final step, you have to image the sample of interest and adjust the PeakForce set point to match the deformation found when scanning the reference sample. So this takes about, in total, half an hour, I would say. For the absolute method, steps 1 and 2 are strictly identical, deflection sensitivity and spring constant. And then instead of using a reference sample, you have to scan a tip check sample. A typical image is shown on the right part of the slide, and get an estimation of the tip radius, which is done automatically by the software. So the absolute method has the benefit that there is no concern over the accuracy of the modulus of the reference sample, or whether it ages over time or becomes contaminated. But on the other hand, you have to image the tip check sample very gently, very carefully, to avoid breaking the tip, because it's a pretty rough and hard sample, stiff sample. So the next slide shows a range of the probes that can be used as a function of the type of samples and their corresponding Young's modulus that have to be imaged, and also the corresponding calibration samples that have to be used. So this list is not exhaustive. This slide is a bit old now, and from that time we have developed 3 other different types of probes at least. For instance, the ScanAssist air probes, which are 0.4 Newton meter and a resonance frequency of 70 kilohertz. Also the ScanAssist fluid probes, which are slightly stiffer, 0.7 Newton per meter, with a resonance frequency of 150 kilohertz, and a typical tip radius, nominal radius of 20 nanometers. We also developed ScanAssist fluid plus probes, which have the same characteristics, but are much sharper, 2 nanometers in tip radius. And as I said a few slides ago, in theory, any type of probe can be used, so then it's up to the user to go and try with his own probes. Moreover, the probes and calibration samples listed here are not suitable for the investigation of soft biological samples like living cells. So I will give you a new list, an updated slide in a few minutes. So there are a few requirements. The sample has to be deformed by at least a few nanometers, or I should better say a few tenths of nanometers. And the cantilever has to be deflected by a few tenths of nanometer too. Simply because if you want to probe properly the mechanical properties, you have to indent a minimum into the sample. You have to deform the sample. The present slide shows one of the first examples we have gotten on DNA. So 3 channels are represented here, though 8 channels can be displayed simultaneously in the nanoscope software. So in that case we only have height, elasticity, or I should say DMT modules maybe, and adhesion. So height scales are respectively from top to bottom 2 nanometers. Then about 1 megapascal for the DMT channel and about 100 piconewtons for the adhesion channel. So you can see a relevant and quantitative contrasts on all the channels, which is remarkable when you think that the DNA strand is less than 2 nanometers in height. So it proves that by using this technique you can apply a very low force and be not too dependent on the scaffold. So it works really well on DNA, most of polymers. Even biological polymers sometimes, like amyloid fibrils or collagen. So talking about amyloid fibrils, if you're interested, we have a nice paper just released in Applied Physics Letters, which I can forward to you, of course. But the question is, okay, it works really well on DNA and polymers, but is it applicable to very soft samples, like biological samples? So before I start answering that question, I would like to show you a last example taken on native membranes this time. So of course, when new technique like this one is released, a question you might come across could be, okay, you can monitor the mechanical properties, but does the accuracy of the mechanical measurements impact the quality of the image? So on that slide is shown a native bilayer of bacteriorhodopsin proteins. So BR is a transmembrane protein, fully transmembrane protein, which acts as a proton pump, capturing the light energy and using it to move protons across the membrane out of the cell. So it typically assembles into 2D patches that can cover approximately 50% of the membrane, and it's found in most of the time archaeal cells. So they form trimers, and were first imaged in contact mode in the 90s, and then in tapping mode later on. So they were imaged approximately a year ago by using PeakForce Tapping with cantilevers having a spring constant of 0.4 Newton per meter, and you see that the lateral resolution is really good. It's about a few angstroms. I think it's 4 angstroms, and it's even better to what the autos obtained in 2012. So you can clearly see not only the trimers, but also the different subunits, the peptide loops, and by using specific algorithm you can reconstruct a 3D model of the proteins. So starting from this slide, we're going to review a few examples of PeakForce Tapping and PeakForce QNM images achieved on living samples this time. So working on living samples is a real challenge in FM. So we'll start from the easy, stiffest samples and go to the more difficult samples, the softest samples, which are eukaryotic cells. So what we call a frustule is the porous cell wall or the external layer of diatoms. So the frustule is composed of mainly silica, and it's coated with a layer of organic substance, which is called pectin. And this is a fiber which is most commonly found in cell walls of plants, and it also contains several types of polysaccharides. So the frustule structure is usually composed of two overlapping sections known as thecae, and the joint between the two thecae is entered by bands of silica that we call girdle bands that hold them together. The frustule also contains many pores to provide the diatom access to the external environment, which is the seawater, for processes such as waste removal. So the top left image is an electron microscopy image, which is a sample courtesy of Dennis Kunkel from Microscopy Incorporation, where you can clearly see the pores, allowing communications with the external environment. All the rest shown on this slide are AFM images captured by using PeakForce QNM. So the color scales are indicated on the images. The 3D height and PeakForce error channels show a nice resolution. It looks actually like a waffle. The addition is not very informative, but it's not very surprising, because when you operate in liquid environments, you greatly reduce the adhesion forces. So you're not supposed to see a great especially the area which is immediately surrounding the pore. This seems to be much softer and more deformable than the rest of the frustule, which is about 200 kilopascal. So the tip was calibrated before the experiment, so everything is quantitative. And as far as I know, this is the first time that such a sample is imaged by FM. So let's stay in the seawater for a while with the second type of sample, which is a diatom. So diatoms are a major group of algae belonging to the phytoplankton, and most of the time they are unicellular but can exist as colonies too. So it exists about 200 different types of diatoms that can be found in very various environments like the seawater, freshwater, soils, and so on and so forth. And they are very resistant to the external environment factors because of this silica wall. So they have been quite widely explored by FM, but most of the time in topography. With respect to the mechanical properties, there are a few publications, but mainly based on force measurements, most of the time force volume. So we investigated the mechanical properties of living diatoms by PeakForce QNM and found different ranges of Young's modulus for the different parts of the diatoms. So I don't want to go too far into details, too deep, but I can give you a few examples. So for instance, the silica stripes had an average Young's modulus of about 44 megapascal, whereas the matrix between the stripes was 2 times softer, 21 megapascal. Some other areas were found to be much more heterogeneous. From those results was proposed a cell wall model with individual silica nanoparticles incorporated into an organic matrix. Such an organization of girdle band and other structures enables the high flexibility needed for motion and adaptation to different environments while maintaining the integrity of the cell. So if you're interested, that study was just published in Journal of Ecology, which is, I think, the best journal in algae research, and it should be released in the December issue, if I remember correctly. Normally, we might be able to get the cover picture, which is shown on the right part of the slide. So in any case, that study was definitely more informative than any force volume work reported so far. Okay, so now we'll go to softer samples with bacteria, especially E. coli. So E. coli are gram-negative aerobic or anaerobic and non-sporulating bacteria. About the dimension, they are about 2 micrometers in length and about 500 nanometers in width and height, though this can be variable depending on species. They are rather robust bacteria that can live on a wide variety of substrates. They are very resistant. Unlike most of E. coli species, K12 strains are able to multiply in the intestine and are particularly resistant to antibiotics. And one of their other characteristics is that they possess pili, which are shown in figure A, that typically retract under depletion conditions or any stressing environment. And this is one of the reasons why they are extremely hard to image by FM, whatever the technique. So over the last decade, many people tried to image them by using contact mode or tapping mode, but didn't succeed. So as you can see, by using PeakForce Tapping, very good topography images can be obtained in about 20 minutes. So this is the center image, which is shown in 3D. You can see on that image that the pili are already retracted, or you can see maybe some remnants of them because they are under stressing environments. So we calibrated the probe before the experiment and found that the average Young's modulus was about 183 kilopascal, which is in perfect agreement with previous studies based on force measurements. So now we're going towards more difficult samples, living eukaryotic cells, which are even more challenging to image because they are much softer and they move under the tip. So the first example is about glioblastoma cells. Those cells are highly invasive and are found in the vast majority of brain cancers. They are by far the most common and malignant form of brain cancers. And this disease is not so easy to cure, not to say impossible, because first, most of therapeutic agents cannot cross the hematoencephalic barrier, which is rather permeable, and second, because conventional therapy, which means surgery, is very likely to damage the brain, of course. So this is the reason why understanding how this type of cancer is mediated, and being able to detect it at an early stage, if possible, is essential, is so important. So in the following experiment, the cells were doubly transfected with 2 tumor suppressive factors, either TP53 or P10. So all the details of the transfections are shown here. I don't have so much time to comment on them, but if that slide is not clear, I'll be happy to answer your questions at the end of the presentation. So to make it clear, if the cancer cells are successfully transfected with any of the tumor suppressive factor, either TP53 or P10, they are no more invasive. This is what you have to keep in mind. So successfully transfected means that they have to be induced by chemical primer, which is called doxycycline. So we believe that whether the cells are cancerous or not should totally change the mechanical properties. So this is why we imaged all of the cell types, including controls, by using PeakForce QNM. So the next slide gives a representative example of what can be expected in terms of lateral resolution when using PeakForce Tapping or PeakForce QNM. So typically living cells are pretty hard to image in contact mode or tapping mode, and impossible to image by using force volume, except if you want to go for a very low resolution. And by PeakForce Tapping, you can see on the left image the high resolution height image of a living glioblastoma transfected with TP53, one of the tumor suppressive factors. So by applying an average force of 200 or 300 piconewtons, which is pretty low, or let's say reasonable, the probe can sense both surface features and inner features like the nucleus or the cytoskeleton that you can clearly see here. So the sample was not fluorescently labeled in that case, but it's most likely actin cytoskeleton, I would say. Another interesting option offered by the Bruker analysis software is that you can display a 3D rendering of the topography, for instance, and add what we call a skin. So in other words, another channel, and you have the choice between 8 channels, which can be interesting, for instance, to correlate some cellular features with some specific mechanical properties or chemical properties. So in that case, on the right part of the slide, we displayed the deformation skin on the top of topography. So to make it clear, the cytoskeleton fibers, most likely actin fibers, are dark because they are hard to deform, whereas the portions between the fibers are brighter because they are much softer or easier to deform. But the main goal in this type of experiment, of course, is to make statistics out of the AFM data. So in other words, make as many images as possible. So for this we need to generate fast AFM data. So on this slide, are shown low resolution images of glioblastoma, 128 pixels per lines and same number of lines, which can be taken in approximately 5 to 6 minutes by using adaptive scan. Again, the contrast in addition is not very informative for the reasons I mentioned before, but the elasticity and deformation channels provide a beautiful contrast. You can clearly see the structures under the cell membrane, the organelles, and you can clearly associate the mechanical behavior to some cell parts, like the nucleus or the lamellipodia. And the resolution is not perfect, but it's more than good enough to process the data by making a bearing analysis. So I didn't talk about this before, but a bearing analysis means that you can display an histogram or distribution of the color out of any channel, topography, elasticity, adhesion, deformation, whatever you want, and get the average value. So on the next slide, I'll summarize the results of our experiments on glioblastoma. More precisely, the average Young's modulus on the top and the deformation on the bottom for all types of samples of cells. So 2 slides ago I was saying that to be successfully transfected, the cells has to be induced by a chemical agent called doxycycline. This is what IND means on the graphic. IND is induced. So you can see that most of the cell types which are highly invasive, this means all controls including non-induced samples, have a Young's modulus of about 40 kilopascal, whereas the cells transfected by the 2 tumor suppressive factors, TP53 or P10, are much stiffer. So typically the average Young's modulus is increased by factor 3. This makes totally sense because the cells have to be softer and easily deformable if they want to be invasive. And this is pretty nicely correlated with the deformation channel. So the softer the cells, the more deformable they are, and vice versa. So be careful, I'm not saying that PeakForce QNM can be used to detect any type of cancer, but at least in this case it works, and this is very interesting, promising application. So the next sample is focusing on keratinocytes. Keratinocytes are the major components of the outermost layer of the human skin. They are able to make tight junctions with neighbor cells and even with other cell types such as neuronal cells, and represent the main barrier against UV radiations for the human body. So HaCat is a particular cell line of keratinocytes which is immortal, and this one is very widely investigated in cytology, mainly because it's immortal and very close to the human keratinocyte, and also represents a good candidate to explore the potential of PeakForce QNM. So the cells were exposed to glyphosate, which is an oxidative agent that has been shown to have the ability to go through the skin and cause severe impairments. First, control cells were explored by PeakForce QNM, and then different concentrations of glyphosate were tested on living HaCat. So the main consequence is first a striking change in topography. So you can see on the top left image that cells tend to retract and synthesize stress fibrils in less than half an hour. And also a very striking change in elasticity. For a glyphosate concentration of 15 millimolars, the average Young's modulus is increased by a factor 3 approximately. So something we didn't talk about in the previous slides and example is the fact that you can monitor the dissipation between the tip and the sample during each tap and display them in electron volts, or most of the time kilo electron volts. So to convert this into joules, you have to multiply this by the Planck constant. And I think it's very important to mention that AFM data can be easily converted into kinetic parameters. So interestingly, the adhesion image tells us that the adhesion is much more important. You can see brighter spots between the cells than on the cells. And also between the cell and the glass, which is the support, the adhesion is pretty low. Of course, this is non-specific adhesion, but an experiment with functionalized probes and specific antibodies at the end might tell us that or what type of molecules are involved in the cell-to-cell recognition or adhesion. So this might be an interesting idea for a further experiment. So another interesting application of PeakForce QNM. So now I would like to introduce MIRO, which is a microscopy image registration overlay software. That program was developed by vivo to automatically overlay optical and AFM images in a short time. So the principle is the following. You import an optical image, most of the time a nice fluorescence image, inside the AFM software, and use the mouse pointer to define the location where to make the AFM image. So the sample will automatically be moved to that place, to the desired location, and the AFM image is captured and fully integrated inside the optical image. So this is possible in a tool that we call the BioScope Catalyst, which is nothing but an AFM mounted on an inverted optical microscope. And with this, you can combine AFM data with basically any type of optical microscopy, especially fluorescence microscopy techniques. So if you want to have more details about the calibration, the detailed features of MIRO or the compatibility, I can certainly explain this to you offline, but this is not the purpose of the presentation. So on the bottom parts of the slides are shown two examples of overlay. So this, the one on the left was recorded on living HeLa cells. So the nucleus is in blue, the actin cytoskeleton in red at the periphery of the cell, and the tubulin in green. So we used this MIRO software to test two drugs that can disrupt specifically actin or tubulin cytoskeleton. So those two drugs were latranculin and nocodazole, and we used MIRO to select the best locations where to perform the force curves. So we could see that when destroying the tubulin cytoskeleton, the cell stiffness was changed by about 10 to 20%, whereas when destroying the actin cytoskeleton, there was a dramatic change in elasticity, a softening of about 50 or 60%, if I remember correctly. The other example shown on the right part of the slide was recorded on HaCat cells, the same cells that we mentioned in the previous slide. So in that case, the nucleus is in blue, the actin cytoskeleton in green this time, and in red we have not the mitochondria, but the endoplasmic reticulum. So after this explanation, I think you totally understand the power of combining PeakForce Tapping, especially PeakForce QNM with MIRO, especially if you can use fluorescence microscopy. So the example you have on the present slide was acquired on UVEC cells from umbilical cord and shows how easy it is to overlay PeakForce QNM images together with fluorescence images. So the AFM image shown in the center is itself a mix of PeakForce channel, which is close to topography, and Young's modulus channel. You can also make that AFM image more or less transparent by playing around with the opacity to show how it fits with the fluorescence image in the background. The individual PeakForce images, PeakForce QNM images, are also shown at the bottom of the slide. So a possible application could be, for instance, add a drug to the dish and probe in real time the effects in the optics and correlate this with specific mechanical properties. Okay, I have to move ahead because we have only 12 minutes left. This slide is actually a summary of all the samples that have been tested so far, explored so far with this technique, and a choice of recommended probes. So the scan-assist fluid probes I mentioned before can cover a very wide range of applications, but should not be used for eukaryotic cells that are too soft. I mean below 50 kilopascal, because they are too stiff. They're about 0.7 Newton per meter, which is quite stiff for eukaryotic cells. On the other hand, some eukaryotic cells like neurons or hepatocytes can be really soft, a few tenths of Pascal, not kilopascal but Pascal, and in that case the OBLB probes can be used to probe them. So the OBLB is actually, as far as I know, the softest existing probe on the market. It's 6 millinewton per meter, which is really soft. I recommend OBLB and not OBLA because although it's very short, it's too short although it's very soft, it's too short to probe most of the living cells. And to fill the gap in between, we developed prototype probes I cannot name, but at least I gave you the spring constant in the inset on the top of the figure. So to be really quantitative, one has to properly calibrate the probe. And none of the calibration samples provided in the kit can be used. Even the low density PDMS would be too stiff to probe any type of eukaryotic cell. Nevertheless, I can recommend gelatin or agar samples, which are most of the time homemade samples, with which you can go down to 1 kilopascal in terms of Young's modulus. So I have to say that the technique is not perfect, and to be fully quantitative, especially on super soft cells, we might have to design new probes with a low spring constant and a larger tip radius to have a better distribution of the force and a better control on the loading force. So the key to success for this type of experiments is really the quality of the force curves, which is mainly obtained by working on the PeakForce set point and also the scan rate. So typically, if you want to have really good force curves, you should scan slow, pretty slow. So if you want to have additional information on the scanning parameters, just let me know. I'll be happy to help out. So the next slide is about the conclusion. So since its development, PeakForce Tapping or PeakForce QNM have greatly improved to extend the range on biological samples. Though it's still not 100% quantitative for the softer samples especially, a very wide range of applications can be covered now. But again, it's not perfect. Working on super soft samples requires an accurate calibration, which is very difficult to obtain on agarose and gelatin. Also, I must say that so far Scan Assist works really well on most of the samples, but not very well on soft cells. So it's better to select in that case individual and turn off the auto set point. The rest, which means auto scan rate and autogen works really well. So this can be turned on, no problem. So we're still working on improving the technique, which is already good, which already gave excellent results on living cells. So I think you have pretty good examples in that presentation. Also, PeakForce QNM might work with functionalized probes and might offer very interesting perspectives, but this requires further testing and maybe further development. So next week will be held the 4th edition of the FM Biomed in Paris. We chose this event to release an application note that summarizes all that has been said in this presentation and slightly more. So I guess the PDF version will be downloadable from the Bruker website very soon, in a few weeks. And what you can see here on the slide is the first page of the application notes. So the bubble images were on the top were kindly provided by some of my collaborators. So talking about collaborators, I would like to thank greatly the following persons for having provided great samples to analyze. It really helped a lot testing PeakForce QNM and defining more clearly its potential and limitations. And in some cases we could publish an article, so some of them are still under submission, some of them are not written. So I think those people also benefited from this collaboration, hopefully. So first, all people from Zagreb, Croatia, for the work on diatoms, especially Vesna and Galia. Gregory Francius from Nancy for providing the E. coli samples, E. coli K12. All people from the Hochschule Mannheim, Physiology Institute in Mannheim, to have provided many different cell types like fibroblasts, HeLa cells, UVEX, glioblastoma, neurons. This is our oldest collaboration actually. And at last I would like to mention the latest collaboration from Besançon in France for the work on human keratinocytes. So thanks to all of them. So this is my last slide about the contact information. If you have any questions, feel free to contact me anytime by email or phone. So the first number is a mobile phone number and the second one is my landline. We also have the Bruker Nanoscale World Forum, which is probably the best way to communicate or exchange ideas, not specifically on PeakForce Tapping or PeakForce QNM, but any topic related to the FM world. It's not only for FM experts. You can be an FM beginner, or you can have never tried FM before. This is not a problem. We'll be happy to help you. Also, John kindly provided very interesting web links about PeakForce QNM, about Scan Assist, about the BioScope Catalyst. I also would like to mention, as I was saying at the beginning of the presentation, there is an exit survey, so please take a few minutes of your time to fill it in, and it will help us a lot improving the quality of our webinar presentations in the future. So thanks a lot for your attention, and thanks to John for his technical assistance. Okay, so I would like to go to the questions now. Okay, the first question I see is any other models, such as Sneddon's model, available to calculate the modulus? So that's a very good question. Actually, so far we only have the DMT fit to calculate the Young's modulus, because first, originally PeakForce Tapping or PeakForce QNM were not developed to work on biological samples, but more on a wider range of samples, including polymers and stiff samples. But it appeared that it worked quite well on biological samples. The second reason is I think the DMT model is quite widely used for many nano indentation works. This is the most commonly used. So I think this is the main reason why we choose the DMT model. But I have to say that for most of the studies on living cells, of course I used the raw AFM data, so recorded by using a DMT feed, but also I use an external program called Rambo. So with that program you can first capture some force curves in the Young's modulus image. You have to make what we call a high-speed data capture, which means that on a line you capture typically at a very high resolution a few thousands of force curves. You can define the segment you want to analyze, export those curves in the program, and then you have the choice between I think 4 or 5 different models, including the Sneddon model. So this is what I used for the work on bacteria, on glioblastoma, and most of the eukaryotic cells. So now I would like John to help me, because actually I can see the questions but in a very, very tiny window, so I would like to know if there's any way to undock this. Make it bigger. Oh, okay, I found out.
There we go. Okay.
Yeah, thank you, John. So I will throw the first one to the bin. Oh, another question is, can your software give any information about the influence of the underlying stiff substrate like the glass, since you might measure this influence on the thinner parts of biological samples like the leading edges of the cells? Yeah, that's a very good point. So typically if you indent by a few tenths of nanometers, so on soft samples I would say it's something rather like 100 nanometers or 200 nanometers. So if you're on the very edge of the cell, of course this can be a problem, because in that case you will have an influence of the substrate. You can see this in the last MIRO slide of my presentation. So if you're on the center of the cell, for instance, if you focus on the Young's modulus channel, the contrast is very dark, because in that case you're really specific, and when you get on the edge of the cell it's becoming much brighter, because in that case, of course, the cell is very thin, less than 100 nanometers, maybe 50 nanometers. So this is why it's so useful to use the bearing analysis function, because in that case you can draw a square on the center of the cell and then get the real Young's modulus. You can do the same with the Rambo program, of course. So to answer your question, yes, this is very important, and you have to be careful with this. So we have tons of questions. Sorry, some questions are a bit hard to understand. How do you fix the cells on the solid support? That's another good point. So typically when I work on living cells, I don't use any coating on the dish. Of course you could use polylysine. This would be a good way to image the cells, but typically I just image the cells on the glass bottom petri dishes without any agent. What is MIRO again? So MIRO is an overlay software to overlap AFM and optical data. So of course your AFM has to be connected to an inverted optical microscope, and in that case, after a quick calibration, you can import an optical image and then overlap this image with any AFM channel you want to. So if you're interested, we have an application note about MIRO, a more detailed technical description, and also a few publications. I can forward this to you. Just send me an email. Okay, what is the range of the PeakForce error, which was shown from PeakForce error map during the scanning? I don't know what image you are referring to, but most of the time it's between 100 and 500 piconewtons. So you have to be more specific if you want me to answer that question. Sorry. how to set and adjust the PeakForce amplitude during imaging soft biological samples in fluid. So this is really uneasy actually. You have to first find the right probe. If the cell is not too soft, you can certainly try with ScanSys fluid probe. And first you need to engage on the stiff part of the sample. You can click Auto Config and then come back to the cell and zoom out. So this is a first point. Then you have to first reduce the PeakForce set point until you detach, and then go back to the previous value, indent a bit into the sample until you get a good tracking. So you have to be careful not to put the gain too high, because this is very likely to give you bad force curves and generate a lot of noise. So you have several tricks like this, but it's a bit hard to answer that question in a few minutes. So you can contact me maybe, or we can discuss it by phone if you want. So it's more than an hour now, and I think I have to stop talking. But again, don't worry about this. We'll collect all the questions, and I will make a summary, generate a Word document, and send it to the field, to all the attendees. So thanks a lot for your attention again, and thanks John for your help. Bye bye.